Hello my dear students, these days we were discussing about some experiment which is mentioned in the chapter Molecular Basis of Inheritance. So we have seen Hershey and Chase, they proved that DNA is the genetic material in the year of 1952. Then after, again, among the scientists, they had a great doubt about the DNA replication. We know that the word meaning of replication is getting a copy. Okay, DNA also can produce a copy in the process called DNA replication. But their doubt was how this replication will take place. DNA, what kind of method it will follow for the replication. So, three types of method put forward by different group of scientists. They were, first one, conservative method. According to conservative method, they are telling that one parental DNA will be there like this. When this parental DNA go through the replication process, it will give two copies of parental DNA. Among the two copy, one will be exactly same as the parent, and second will be newly synthesized one. One will be exactly same as the parent and the second will be newly synthesized one. It was the conservative method. And second, it was semi-conservative method. According to semi-conservative method, there also parental DNA, we can see here like this. These are DNA strands. Okay. Replication will take place, they will give the two copy. According to semi-conservative method, these parental strand divide. One strand here and one strand will be here. So a newly synthesized strand will come and join with the parental strand like this. It was the concept of semi-conservative method. And one more concept was there, that is third one, dispersive method. Third one is dispersive. According to dispersive method, the parental strand, when it go through the replication process, so it will break down into small segments like this. It will break down into small pieces. So in between two segments, a newly synthesized segment will come and join the gap. Like this. All the gap of that segments has to be filled by a newly synthesized DNA molecule. It was the concept of dispersive method. But the two scientists, MESELSON and STAHL, they followed the second concept, which is semi-conservative method. They could prove that DNA always replicate through the semi-conservative method. In order to prove their idea or concept, they performed some experiment in the laboratory condition. So in order to perform the experiment in the laboratory condition, they have chosen an experimental animal, a microorganism, that is none other than E. coli or Escherichia coli. So there were two reasons why they have chosen this E. coli or Escherichia coli for their experiment. First reason, as we know that E. coli is considered as the harmless bacteria. It will not bring any kind of problem, harmless bacteria. And second reason, when the replication take place, it will give a replication in very less duration, which means exactly 20 minutes it will replicate. That is the two reasons they have chosen E. coli as their experimental animal to prove that DNA replication will take place through the semi-conservative method. Okay, in order to conduct their experiment, they first of all prepared two medium like this, and they have given the name number 1 and number 2. Okay, inside the medium they put NH4Cl. We know that is ammonium chloride. Now the medium contain ammonium chloride. So in the ammonium chloride itself we can see the presence of nitrogen. Okay, in the first medium they introduce heavy isotope of nitrogen. Okay, a heavy isotope of nitrogen we can denote with the capital letter N15. So now we can call the first medium N15 medium, which means This ammonium chloride contain heavy isotope of nitrogen. That is why it got the name N15. But here, light isotope of nitrogen they implemented or introduced there. It was N14. We can denote with the letter N14. So the medium name also changed, N15 medium and N14 medium, as heavy isotope and light isotopes respectively. After that, they introduced microorganisms like E. coli into the first cell. So after 20 minutes, E. coli now it is here. Okay, after 20 minutes, they transfer to the next cell or next medium, N14 medium. Then after again they take the result, they observe that there we have seen after replication the strand, parental strand was there like this, a newly synthesized strand came and formed like this, which means they found a hybrid form. Hybrid means two different things come together and make a new one. Old form and new form. This blue is indicating the old form and the green is indicating the new form of DNA strands. So they considered it was the F1 generation. Okay, so then after it particular DNA molecule extracted, then after it dissolved it with the cesium chloride, CSCl, cesium chloride. After dissolving it with the cesium chloride or CaCl, and then taken for the centrifugation process. So we know that whenever the centrifugation will take place, we can get the result in a tube, mainly in two ways. First one is pellet form, which is formed at the or settled at the bottom, and supernatant form that will be settled at the surface. Consider this is the centrifuge tube. After centrifugation, they got some pellet here and supernatant there. They found that the pellet form it was N15 and the supernatant it was N14. But one more thing they found here, in between N15 and N14, at the middle, a hybrid form also they found here, and they have given the name for the hybrid form N14, N15. So they have given the hybrid form for N14, N15. Here also some change took place. The pellet, now the name is N15, N15. The supernatant, now the name is N14, N14. It was their result. Okay, so again I am going to explain what are the changes took place in the DNA, especially when they were in the first or N15 medium and N14 medium. What are the changes took place in the DNA? I will show. So consider this is the parental DNA. Okay, suppose this is a parental DNA. Then it has to be taken to the N15 medium. A replication took place. Okay. Then after it transferred to the N14 medium. Now the DNA is in N14 medium. One more thing you have to know. As long as this DNA in N15 medium, it is known as N15 DNA. Then as long as it is spent in the N14 medium, it is known as N14 DNA. Now it is N14 DNA. Then the stranded, actually according to the rule of semi-conservative method, DNA is act as a template. Okay, now the DNA strand divided into two and act as a template here. Into the template, newly synthesized DNA strand will come and join like this. Okay, so they got a hybrid form at F1 generation. So always you have to remember and remind the timing or duration, only 20 minutes. Within 20 minutes they got the result. First form or F1 form they got. Then they waited again next 20 minutes. Which means 20 plus 20, 40 minutes for the F2 generation. So in the F2 generation, they have chosen the F1 generation for the next 20 minutes. After 40 minutes, they found that now still it is in the N14 medium. So these are the N14 DNA. So let me draw once again here. So this is the F1. Okay, this is the F1. After 40 minutes in the N14 medium, again these strands divided. From here we got 2 strands independently, we got 2 strands here also. Then what happened? Now it is in the F14 medium, newly synthesized DNA strand came and joined with this one. So in order to show that newly synthesized, as long as it is in the F14 medium, I am using the green color. Here also newly synthesized DNA strand came and joined with the, here also newly synthesized DNA strand, and here also newly synthesized DNA strand, and here also new strand. So we can see here, this one and this one is a hybrid form. 2 hybrid form we can see at the same time. Because why it is known as hybrid? N15, N14 here. This blue stands for N15, here also N15, and all the green stands for N14. This is N14, N14, this also N14, N14, N14. So here we can see that 2 hybrid form and 2 N14 also we got. It was the F2 generation result. Hence they prove that DNA will replicate, which means it will act as a template according to the method of semi-conservative method, which is followed by these 2 scientists, MESELSON and Stahl. Okay, they prove that DNA will replicate or will make the copy. In the replication time, DNA will act as the template and the strand will divide, and newly synthesized strand will come and the join will give a hybrid form. It was the concept which is proved by MESELSON and Stahl in the year of 1958. I hope you understood. Thank you.